Tissue engineering has long depended on geometrically static scaffolds seeded with cells in the lab to create new tissues and even organs. The scaffolding material, usually a biodegradable polymer structure, is supplied with cells, and the cells, if supplied with the right nutrients, then develop into tissue as the underlying scaffold biodegrades. But this model ignores the extraordinarily dynamic morphological processes that underlie the natural development of tissues. Now, researchers at the University of Illinois, Chicago have developed the new 4D hydrogels, that is 3D materials that have the ability to change shape over time in response to stimuli, that can morph multiple times in a pre-programmed or on-demand manner in response to external trigger signals. In a new study published in the journal Advanced Science, the UIC researchers showed that these new materials may be used to help develop tissues that more closely resemble their natural counterparts, which are subject to forces that drive movement during their formation. The hydrogels can be programmed or induced to undergo multiple controllable shape changes over time. This strategy creates experimental conditions to partially mimic or stimulate the continuous different shape changes that developing or healing tissues undergo. And it may let the researchers study morphogenesis and also help them engineer tissue architectures that more closely resemble native tissues. The novel material is made up of different hydrogels that swell or shrink at different rates and extends in response to water or the concentration of calcium. By creating complex layering patterns, the researchers can guide the conglomerate material to bend one way or another as the layers swell and or shrink. The researchers say they can change the shape of these materials by adjusting, for example, the amount of calcium present. In their experiments, the researchers were able to cause the hydrogel to form into packets similar in shape to alveoli, the tiny sac-like structures in the lung where gas exchange takes place. Not only are these hydrogels able to change their architecture multiple times, but they also are highly cytocompatible, which means they can have incorporated cells and the cells remain alive, something that many existing 4D materials are unable to do.